In the pristine waters of Antarctica, where human influence seems most remote, scientists have made a discovery that could transform our fight against one of the most pervasive pollutants on Earth. A newly discovered bacterium with an appetite for plastic has emerged from the icy depths, offering unexpected hope in our struggle against microplastic pollution. Today, we dive into this remarkable story of scientific serendipity and explore how a microscopic organism from Earth's southernmost continent might help solve a global crisis. In March 2023, a team of researchers led by marine microbiologist Dr. Elena Kowalski made a startling discovery while collecting samples from McMurdo Sound, Antarctica. The joint expedition between the University of Wollongong and the Chilean Antarctic Institute wasn't specifically looking for solutions to plastic pollution. They were primarily investigating extremophile adaptations in Antarctic marine ecosystems when they noticed something unusual: bacterial colonies forming on plastic debris collected during their sampling. This bacterium, now tentatively named Pseudomonas Antarctica MP1, was doing something remarkable. It was consuming microplastic particles in water temperatures between 1 and 4 degrees Celsius, conditions previously thought to severely inhibit such biodegradation processes. Initial genetic sequencing confirmed they had found something entirely new, a novel strain with significant divergence from known Pseudomonas species, suggesting specialized evolutionary adaptations to the extreme Antarctic environment. What makes this discovery truly groundbreaking is the bacterium's efficiency. Laboratory tests showed that these microorganisms could reduce the mass of standard microplastic test samples by approximately 13% over just 60 days, a rate considerably faster than any previously documented cold water biodegradation. Further analysis revealed the secret to this ability: novel enzymes named PETase-K1 and MHETase-K2 that enable the bacterium to break the chemical bonds in polyethylene terephthalate, or PET, polymers. Unlike previously identified plastic-degrading enzymes that require temperatures above 30 degrees Celsius to function optimally, these enzymes demonstrate peak activity at just 4 degrees Celsius. This represents an entirely new class of cold-active biocatalysts, evolved specifically for function in one of Earth's most extreme environments. The bacteria don't just break down the plastic. They metabolize it, using the carbon compounds as an energy source, essentially converting our pollution into their food source. The discovery quickly captured attention beyond the scientific community. By September 2023, environmental engineers at the Swiss Federal Institute of Aquatic Science and Technology, known as EOAG, recognized its enormous potential for water treatment applications. A collaborative project codenamed CryoFilter was established to develop a biofiltration system using immobilized colonies of the Antarctic bacteria. The prototype, currently being tested at a wastewater treatment facility near Zürich, incorporates a revolutionary membrane design where the bacteria are embedded in a hydrogel matrix. Dr. Markus Holzner, the project's lead engineer, explains the dual function of this system. The bacteria essentially function as both a physical filter and a biological degradation system. They produce a sticky extracellular matrix that captures the particles and slowly breaks them down over time. What makes this approach revolutionary is that the filter becomes more effective with use rather than requiring frequent replacement. Initial results are promising. The system can capture and begin degrading up to 87% of microplastic particles in the 10 to 100 micrometer range, precisely the size class that conventional filtration systems struggle to address effectively. Developing the CryoFilter system wasn't without significant challenges. The engineering team faced a major hurdle: maintaining bacterial viability within the filtration system. These microorganisms evolved for specific Antarctic conditions, so keeping them active in a treatment facility required innovative thinking. The solution came through biomimicry, specifically, a design inspired by Antarctic sea ice microbial communities. The bacteria are suspended in a cryoprotectant hydrogel containing glycerol and specialized polysaccharides that mimic the microenvironments found in sea ice brine channels. This innovation allows the bacterial colonies to remain metabolically active for up to 6 months without replacement, dramatically improving the system's economic feasibility. The CryoFilter also incorporates sophisticated monitoring technology using fluorescence spectroscopy to track bacterial activity and degradation rates in real time. This allows for automated adjustment of flow rates and nutrient delivery to optimize performance across varying conditions, ensuring the system maintains peak efficiency even as environmental factors change. This breakthrough comes at a critical time in our battle against plastic pollution. A January 2024 assessment published in the Environmental Science and Technology Journal estimated that the average human now ingests approximately 5 grams of microplastics weekly, equivalent to consuming a credit card. Marine ecosystems face even more severe impacts, with microplastic concentrations in some ocean gyres reaching 200,000 particles per square kilometer. These statistics highlight the urgency of finding effective solutions to microplastic pollution. Conventional water treatment plants simply weren't designed to filter out these microscopic plastic particles, allowing them to pass through into our drinking water and natural waterways. The CryoFilter system targets precisely this gap in our current infrastructure, offering a potential solution to one of the most persistent forms of pollution on our planet. As plastic production continues to increase globally, the need for remediation technologies becomes increasingly critical. Despite the promise of this technology, significant challenges remain before the bacterial filtration system can be scaled for widespread use. Current prototypes can process approximately 5,000 liters of water daily, far below the millions of liters processed by conventional treatment plants. Engineers are exploring modular designs that could scale the technology for industrial applications while maintaining efficiency. Perhaps more importantly, the research team is addressing concerns about potential ecological impacts if the modified bacteria escaped into natural waterways. They're engineering biological safeguards, including metabolic dependencies, that would prevent the bacteria from surviving outside controlled environments. Dr. Sarah Johansson, an ecotoxicologist at Stockholm University not involved in the research, has raised important questions about long-term implications. While this technology shows tremendous promise, we need comprehensive studies on the degradation byproducts and their potential toxicity. Previous research has shown that partial plastic degradation can sometimes produce compounds more bioavailable and potentially more harmful than the original polymers. To address these legitimate concerns, the research consortium has established an independent ecological assessment panel that includes representatives from 5 countries' environmental protection agencies, overseeing comprehensive testing programs examining everything from byproduct toxicity to potential horizontal gene transfer risks. Perhaps the most surprising development in this story has been the potential economic value of the degradation byproducts. The bacterial metabolism of PET produces terephthalic acid and ethylene glycol, both valuable chemical precursors used in various industrial processes. Preliminary calculations suggest that at scale, the recovery of these compounds could offset approximately 30% of the system's operational costs. This unexpected economic advantage has attracted investment from major chemical manufacturing firms, including Germany's BASF and Japan's Mitsubishi Chemical Holdings, both of which announced funding commitments to the project in February 2024. Their involvement could significantly accelerate the technology's development timeline. As Dr. Kowalski notes, we initially approached this as purely an environmental remediation technology. The realization that it could simultaneously function as a chemical recovery system was completely unexpected. It transforms the economic equation for adoption. The recovered monomers have been successfully incorporated into a circular manufacturing process that produces new PET products with up to 23% recycled content, a significant advancement over conventional mechanical recycling methods, which typically degrade polymer quality with each cycle. With the technology showing such promise, implementation plans are already underway. Pilot installations are scheduled for coastal treatment facilities in Chile, Australia, and Norway by early 2025. These locations were strategically chosen to test the system across different environmental conditions and types of plastic pollution. The research consortium has developed an open-source licensing model that will make the core technology available to municipal water authorities worldwide while maintaining commercial applications for industrial-scale implementations. This hybrid approach aims to maximize global access while ensuring continued research funding. The technology's potential extends beyond traditional water treatment facilities. Researchers are already exploring applications for marine deployment in particularly affected areas such as coastlines and river mouths. Prototype floating bioreactors containing the bacterial filtration system have shown promise in preliminary tests in controlled aquatic environments. These could potentially be deployed to address plastic pollution at its entry points into the marine ecosystem, preventing wider dispersion of microplastics into ocean currents. The story of Pseudomonas Antarctica MP1 represents a powerful example of how fundamental research in extreme environments can yield unexpected solutions to global challenges. As Dr. Kowalski reflects on the journey from discovery to application, nature has been solving complex problems for billions of years. This bacterium evolved in one of Earth's most extreme environments to utilize whatever carbon sources were available, including our plastic waste. The elegance of the solution reminds us that some of our most pressing environmental challenges might find their answers in the planet's most remote corners. This bacterial species evolved in isolation, adapting to one of Earth's harshest environments. Yet it may hold the key to addressing a pollution problem of our own making. The irony isn't lost on the research team that in our most pristine, untouched environments, nature has developed tools to remediate our most persistent pollutants. As the technology advances toward commercial implementation, it represents how nature's adaptive capacity might help us address the very problems our technology has created. As we look to the future, the implications of this discovery extend far beyond the current applications. The enzymes identified in Pseudomonas Antarctica MP1 could potentially be engineered for even greater efficiency or adapted to target other types of plastic polymers beyond PET. Genetic analysis of these novel enzymes provides templates that could revolutionize how we approach biodegradation of synthetic materials. While the CryoFilter system represents an important step forward, it's just the beginning of what could become an entirely new field of bioremediation. The research team is already exploring potential applications beyond water treatment, from soil remediation to marine cleanup operations. There's even preliminary research into incorporating similar enzymes into textile manufacturing processes to create inherently biodegradable synthetic fabrics. Perhaps most importantly, this discovery reminds us that as we face growing environmental challenges, we should look not only to our most advanced technologies but also to the natural world for solutions. The planet has been conducting its own research and development for billions of years, evolving organisms capable of extraordinary feats of chemistry and adaptation. As this Antarctic bacterium demonstrates, sometimes the most elegant solutions to our most complex problems may already exist in nature's vast laboratory. The discovery of Pseudomonas Antarctica MP1 represents a remarkable convergence of scientific serendipity, evolutionary biology, and environmental engineering. From the frigid waters of Antarctica to wastewater treatment plants around the world, this microscopic organism offers new hope in our battle against microplastic pollution. The development of the CryoFilter system demonstrates how nature's innovations can be harnessed to address human-created problems, potentially transforming plastic waste from persistent pollutant to valuable resource. While significant challenges remain in scaling and implementing this technology, the collaborative efforts of scientists, engineers, and industry partners show promising progress toward practical solutions. As pilot implementations prepare to launch in coastal facilities across 3 continents, we're witnessing the evolution of an idea from laboratory curiosity to global environmental solution. Perhaps most importantly, this story reminds us that in nature's most extreme environments, in organisms invisible to the naked eye, we may find answers to some of our most pressing environmental challenges. The plastic-eating bacteria of Antarctica stands as testimony to both the remarkable adaptability of life on Earth and humanity's capacity to learn from and work with nature's elegant solutions. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.